Hello, this is Brian Kinghorn and I'm going to tell you about using pre-assigned groups under mixed mating groups. You'll have seen in the main video on mixed mating groups that MateCell controls the allocation of individuals into these groups in order to optimize the objective function. You may not want this. You may want to have pre-assigned groups so that you constrain which individuals can be candidates for each of the mixed mating groups. For example, here in mixed mating group 3, you can see that all the males come from pre-assigned group Borstud 2. I'm also going to tell you about shared grouping, and for that, for example, you can see that individuals in pre-assigned group Borstud 1 can be shared across mixed mating group 1 and 2. And you can also see that individuals belonging to group 0 can compete to enter any mixed mating group. So with shared grouping, I'm going to give an example to do with multi-sire joining in beef, and also an example of control of flow of genetic material across breeding units in pigs. This is about the most simple demonstration of using pre-assigned groups. Compared to the main video before without grouping, what we've done here is just added a few lines to the file mixedmatinggroups.ini. Now you'll see we've got 3 mixed mating groups in this example. So because I have put in here a 0 for sequential numbering in the main data file, that 0 says to MateCell I'm using sequential numbering in the main data file. Let's have a look and see what that looks like. Here's the data file, and it's simply adapted from our MateCell Classic demo file. Here are a bunch of females, and you can see that I've simply allocated these to groups 1, 2, and 3, and the same for males. These max uses for males of 25 will be taken as 1, because as you noted from the previous video, max use is only used to signify candidacy or not. Now you'll see that I've allocated group 0 to a bunch of males and females here, and that means that they can be allocated to any of the 3 mixed mating groups. You'll also see that I've used must use equals 1 for a bunch of individuals, male and female, and that means simply that they have to be part of the solution in the analysis. So let's look at the run. I've just opened the file and we'll make the run. And if we look at the top of the console, we can see first of all the numbers in the male and female groups. That's the groups in your data file and the max uses of those. And then you can see the requirements for the mixed mating groups, including the number of candidates that are available for each of these, which in this case, because there's a one-to-one relationship between pre-assigned group and mixed mating group, 12, 13 and 7 is the same as 12, 13 and 7. Plus there are ungrouped candidates which are available. Now this is useful where you have more involved analyses and MateCell may not be able to achieve what you have asked for. This is where you look to find out how to remedy those things. So let's resume the analysis and look at the mate selections. So now you can see that the pre-assigned groups 1, 2 and 3 are allocated into the mixed mating groups 1, 2 and 3 on a one-to-one basis in this simple analysis. And of course there are some individuals that have been allocated to these groups for optimality, but they were free to go anywhere, for such as Leighton and Cohen over here. And these 2 individuals had to be used because must use was set to 1. The must use individuals will always be at the top of the lists in their respective mixed mating groups. Before moving on to shared mating groups, it's useful to put names to these mating groups because they are going to be shared across mixed mating groups. And that's quite easily done. So far we had a 0 in this position here, just to signify that we had sequential numbering already in the data set. What we can do is use the same sort of format as we used in impgroups.txt for normal allocation of mates and MateCell. And here we are not doing any shared grouping. That would come with an extra paragraph, as you'll see in a moment. But because we've got 3 male groups and 3 female groups, these are going to be allocated to the 3 mixed mating groups in sequence. All we've done now is given a mechanism to put names to these groups and also to allow subgroups. So for example, if we had another group in our data set, say group number 10, and it had AI bulls in it which came from a different source, but you wanted to treat them exactly the same as group 1, then you would say we've got 2 subgroups, and the 2 subgroups in the data set are 1 and 10, and these will both be treated as group 1 for the purposes of allocating to our mixed mating groups. However, I'm not doing that, so I shall revert to what I had before, and we can carry on and do the run. And I'll pause that just to show you that we've got exactly the same layout as we had before. We haven't changed anything except that we've added these names to the mating groups, and that's going to make it useful not so much for this run, which is quite simple, but for future runs where we're going to be using shared grouping. And now you can see we've allocated names rather than numbers to the different groupings, and that's going to be useful when we go to shared grouping right now. My first shared grouping example is multi-sire joining in beef, just as in this slide from the main video on mixed mating groups, where we're going to put a number of groups of males in with a number of groups of females. And that number is 7. We're going for 7 mixed mating groups. And you can see the shared grouping aspect with the AI bull category of animals being shared across the first 3 mixed mating groups. And just as in the previous example I showed you, we're using the same data set, and we have 3 categories of males and 3 categories of females, and they are listed under a mating group in the data file. And you can see that the user has good flexibility to match different male groups with different female groups in making up the mixed mating group allocations. So in this example, we've got 3 groups, AI bulls, and of course with controlled mating in bulls, you have single sire joining possible. Now this is not necessarily sensible. What you would normally do is run your AI breeding program up front, and once those decisions are made, they can be included in committed matings, and then run the rest in mixed mating groups for cleanup or backup matings, or indeed for other main round matings. So this example is not necessarily sensible, but it is illustrative. Down here we can see that the paddock sizes are allocating so many females from cows and old cows into 3 paddocks, with this number of natural mating bulls to be selected to go into these 3 groups. And the last category I've got here is female semen. That is egg-bearing semen from bulls. Now that is again not necessarily sensible, but it is illustrative. Now these 3 bulls, you could have them already lined up, or you could use mixed mating groups to select them out of a pool of males. And if you're using egg-bearing semen, probably over old cows with good maternal information available, you're aiming the 7th mixed mating group to produce heifer progeny. So what you would do with these old cows is in the parameter file you'd put extra emphasis on maternal traits to help create excellent heifers to come out of this mixed mating group. So push these MMG 7 females on maternal traits. And of course any animals in group 0 in the main data file are free to go into any mixed mating group. So I'll do a demonstration of a simplified version of this scenario, and it's simplified in that because I'm using MateCell Classic Demo, which is not a very large data set, I'm just going to use for simplicity 4 males and 12 females per group. And here we have our scenario. It's the same as you've seen before, with the same mating groups declared in the data file, but here are our declared shared grouping desires for the 7 mixed mating groups. So this is the thing that you have to add at the end, is the shared grouping information so that you can constrain your mixed mating groups to be constructed as you want. Now for this run, I'm actually going to switch grouping off to start with in both males and females to make a certain illustration. So when we do this, I've just started the job. We can see we do have the male and female groups as in the data file, and we have our 7 mixed mating groups declared, but we just have a simple output in terms of all the numbers because we're not actually applying grouping at this stage. So let's complete the run and print matings. So first of all, you'll see that there is no correlation between the mating group as allocated in the data file and the mixed mating group for males or females. And that's because we're not constraining into the different groupings. Now, MateCell will get a better result according to the other aspects of the objective function because you're not making that constraint. Let's see what happens if we now go and put male grouping constraints on, and I save that and update. Immediately you'll see there's some compromise in the frontier, and it's just evolving back to a position where it gets the best result given the constraint of having males conform to your grouping constraints. And here we can see that we have AI bulls in the first 3 mixed mating groups, except for the second mixed mating group is made up of group 0 individuals that could have been allocated anywhere. And it may well be that these individuals tend to be related to each other and therefore are ending up in the same group to minimize the difference between the 2 points here, to minimize the impact of variation in male reproductive success. Then we have natural mating bulls for the next three groups, as we have here, and female semen bulls for the last group. And now we can go and add female groupings, save that and update. And notice here that we have a compromised initial solution and it evolves back to as good as it can get. Let's print matings and have a look at the females. And here we can see there's confirmation to your grouping constraints of heifers, heifers, cows, 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 old cows and old cows, according to this here. Now you'll recall that in mixed mating group 7 we're using female semen from bulls which are going to be high in maternal characteristics, and therefore we want to select into this mixed mating group old cows that are also high in EBVs from maternal traits in order to specialize in heifer production from this mixed mating group. So we can do that by going to the data set and finding the traits which are appropriate to give extra push for those traits. Now I don't have any of those in this case because this is just simply MateCell Classic demo, but let's say we want to push this trait, back fat, a bit harder. If I save that, I'll look at the out summary file. So without that addition, we have a result in mixed mating group 7 of 286 for that trait. So if I save that and update until it evolves, click print matings, go back to out summary, and that 286 has increased and is now 319. Now that of course is a trivially simple example. What we would do is have a complete index. We already have the default index being applied, and this is the extra push that we have for various traits. And what you're basically looking for is the outcome. The weightings are not so critical, it's the actual impact of those. And so you spend some time looking at the out summary file to see that you're getting what you want, as well as looking at any potential compromises in other areas. And while I'm up here, I'll point out that we did use in the pre-release video for mixed mating groups a trait we called age, and we used that to attract heifers into various groups. That's how we managed this issue before having shared grouping. Now it's more powerful to use shared grouping to organise the types of animals you want to go into the different mixed mating groups are controlled in this more simplified manner here. However, there may be some reasons that you want to have a soft constraint in those things and use weightings instead with dummy traits such as age, in which case if you go and resurrect the video from our obsolete or superseded playlist on YouTube, then you can have a look at that and how it operates. So that leads us on to the example of shared grouping for flow control, as we're calling it, or possibly inventory control. A number of groups have started to look at using this, and the examples I'm going to give are rather simplistic and naive but illustrative. This is the flow of males and females in a simple multi-unit pig breeding operation, and if we take off the sexes and combine them, it makes it a little bit simpler to look at. And there are a number of places where selection decisions are made, from the boar stud to the nucleus farm. And this usually takes the form of the nucleus farm requesting a certain number of boars to be sent from the boar stud, and that would be to top up an existing inventory of boars that they've got. Then there's selection of juveniles out of the nucleus farm to go to the test station, males and females. Then after the test, successful off-test individuals are selected back into the nucleus farm and the boar stud. So these are 3 stages where selections are made and yet no mate allocations are made. And that's where cohorting and grouping of animals is important. Mate selections are being made in the nucleus farm for actual mate allocations, and normal MateCell has been used for all of these decisions, but it's more efficient to use mixed mating groups for the 3 selection only phases and normal MateCell for operations in the nucleus farm. Now we can implement these selections simultaneously using mixed mating groups in a fashion like this. So mixed mating group 1 relates to the selections out of the boar stud, but it's not just the males from the boar stud, it's also the females from the nucleus farm that constitute that mixed mating group. So from the boar stud we might have a need for 20 males altogether. Now many of those will already be at the nucleus farm established there, and so they can have a must-use value of 1, and then what is required is a top-up from the boar stud of that inventory of males. Similarly, the nucleus mature females, whether they're gilts or sows, they are probably mostly going to be kept, and so the number of new females required might be only 20, for example, in a week, and that would be the number that would not have must use equals 1. They are going to be selected from the nucleus mature candidates that are young gilts. For this selection, we're looking to get nucleus juveniles into the test station, both males and females, and that will be a new contemporary cohort of males and females going to the test station. And the third class of selection is off-test selections in the third mixed mating group. So males and females are going to be matched with each other, so that those that go to the nucleus farm as females and those that go to the boar stud as males will be cohorted and matched for future mating potentially with each other. There's no mate allocation taking place here. We're lining up the males and females for potential future matings. And of course, if you're on the ball, you realize that males that are young and come up here will be mating with females that are older, so we're not matching right across the board. However, within the breeding line, we are making selections with some consideration of what the mates are going to be like. We could split hairs and divide this down into age groups and have different mixed mating groups according to age group, but I don't think that's going to add very much to the value of the outcome that's going to be had. And there's always value in keeping things as simple as possible, especially in view of the fact that you might have multiple structures like this, either within a breeding line or across different breeding lines, terminal and maternal. And more likely you have a structure which looks something like this. I've assumed operations in US and EU, a number of different types of breeding units with selection and migration of individuals, in this case probably semen going from US to EU, and quarantine being required for any material coming back. But you will find some structure like this for any animal or plant species that you're dealing with. And the objective here is to make runs, probably weekly, which control the selections and migrations across all these units. This would be for a single nucleus breeding line. And of course, in many operations, you make commercial product lines targeted at specific needs. And in addition to using multiple end uses within matings in the nucleus, we can use the mixed mating groups feature to target these in separate MMGs with indices set as required by the range of customers that you've got. Additionally, of course, there's crossbreeding to consider, and there is the possibility of using our genomic mate selection feature, which interoperates with mixed mating groups to help target these things. That might be something for down the track. And of course, there are multipliers that come out of this operation aiming towards commercial production. And I've represented that just simply with a multiplier in Spain that has young boars and gilts coming from test station 3. And of course, these will be selected particularly for the index, which is appropriate for the product requirements coming out of that multiplier. Now we'll move towards a demonstration. You'll notice that there are only 2 boar studs and yet 3 nucleus farms. And when we look at the parameter file for this example, you'll see that's reflected in the fact there are only 9 male groups and 10 female groups. We actually have 13 mixed mating groups for this example, and that's going to be easier to see when I put that on top of the diagram. So you can see the shared grouping aspect with boar stud 2 being used across mixed mating groups 3, 4 and 5. So let's look at that. First of all, boar stud 2 and nucleus 2 mature females. Now the males boar stud 2 is not exactly just males in boar stud 2. There are 3 categories of males involved in boar stud 2. First of all, there are the boars that are available from migration from boar stud to the nucleus. And then also included in boar stud 2 are the boars that are standing at the nucleus already. 2 categories of those that you deem you want to keep, and you put must use equals 1 for those in the data file, and those that can be competed out from the boar stud, you would use a must use equals 0. And then the number of males that you're going to have coming from the boar stud would typically be a good bit less than, in this case, just 16. You might be only wanting this week to have 2 or 3 of those, and that's controlled by your pattern of must use and the numbers required in the group. So if you wanted 3 boars coming from the boar stud, the number of males with must use would be 13, and that means that there are 3 available to come. You would still have competition if you wanted the standing boars in here to compete with the potential new boars. Otherwise, you would leave them out as non-candidates altogether and cull them yourself, just for age or something like that. So that's boar stud 2 and nucleus 2 mature females, and they are on the EU index because this is the breeding program for Europe. There are 2 other mixed mating groups with boar stud 2. Here they are. and they are different, even although they're all going to be mating with nucleus 3 mature females over here in the nucleus farm. There are two destinations from the progeny out of these matings, that is to test station 3 and back into the breeding operation for Europe, and off to the quarantine station targeting migration to the US. Now, of course, you'd use the US index here and the EU index here, and that's why we have these two separate mixed mating groups. The number going off back for migration to Europe would be typically a lot smaller, and that's reflected in these fictitious numbers that I've put in here. And you'll notice that we're not making mate allocations here, we're just making plans for a grouping of individuals for potential future mating. And the next example here is with off-test 3 males and females. They have two different destinations. One is the ham index going off to Spain for the multiplier, and the other is back into the breeding program with males going back to bore stud 2 and females going to nucleus 3. So when it comes to the example, I'm just using MateCell Classic Demo, so the numbers are not as large as these ones here. And if we go and look at the data file, I've simply allocated mating group using a random number, as you can see, and I've done the same with the must use parameter. So the results that you get won't be typical of what you might expect in something in this, but you will see a sensible pattern of outcome and hopefully how it might relate to you using this in your own operations. So I'll go ahead and make the run. You'll find all these projects under sample data files in your download. So I'll make the run and pause straight away to look at some of the information at the top of the console. We can see we've got the 9 male groups and the 10 female groups and 13 mixed mating groups, and we use the convention MMG for mixed mating group and group for pre-assigned group. Just be careful to be sure you understand the difference between these two things. If we look at mixed mating group 1, the females are nucleus 1 mature females. That's actually female group 4, and that's the only mixed mating group that group of females is used in. So if we come down and look at mixed mating group 1, we need 28 females, and that corresponds to this value of 28 here for the females required per group. That's the group of nucleus 1 mature females. The abs min use for those is 27, and it means that all but one will be pre-selected into that group, and we'll see that in the results. So I'll resume. And the first thing to notice here is that with autocorrect set to 0 in the parameter file, we're optimizing on the result without correction for variation in male reproductive success. And that's because we are going to be making controlled mate allocations in the future, and we don't have the problem of males being aggressive with each other in order to compete for matings. Let's have a look at the matings, print matings, and click. And here we can see the males. Now I've used much smaller group sizes than in the example on the PowerPoint, because this is the MateCell Classic Demo file that's being used as a basis for this. So all the boars will be conforming to the groupings in the parameter file, and you can see that the must uses are at the top and the true selections are those which are zeros underneath them. Let's look at the females. And here's this first mixed mating group where there are 28 females that are to be selected into it, and 27 of them are must uses. So we've only had room for a single new selection of a young gilt probably into this mixed mating group. So that's a simple demonstration. I hope you get a feel for how you might use this in your own operations. You will probably have to spend some time with your own animal or plant species design, working out how you might use this. It's not a simple process, but you may find opportunity to do things that you might otherwise not have been able to do in your breeding operations. The important thing, I think, is that you're making a coordinated analysis across your whole operation, and that gives better control for decisions made in one part of the operation are contingent on what you might make in the other part of the operation, and you've got a more global control over direction for genetic change and traits, and also management of genetic diversity. Of course, in addition to these decisions that you make for selections, you will have mate selection decisions taking place within the nucleus farms or wherever you make those decisions in your operations. And in addition to this, you can take account of other decisions that have been made already in the pipeline with committed matings working with mixed mating groups. Well, that's us coming to the end of the video. You'll need version 17.2 or later to run what you've seen in this video. It's quite new. We're grateful for any feedback and any suggestions for developments that we need to make this more useful for you. Thank you for watching.